to everybody who's attending online. Before I introduce today's speaker, I want to say that the Field Museum acknowledges that it resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. The museum recognizes and is grateful for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. So today's speaker is Dr. Lauren Esposito, and she is the curator and chair of arachnology at the California Academy of Sciences, so in San Francisco. Her research investigates the patterns and processes of evolution in spiders, scorpions, with a focus on tropical islands. Originally from the US-Mexico borderlands, she completed her undergraduate degree at the University of Texas at El Paso and went on to obtain a master's and PhD from the American Museum of Natural History in conjunction with the City University of New York, which is where I did my PhD and how I know Lauren, originally at least. Lauren is the co-founder and director of a science education and conservation nonprofit called Islands and Seas. And she's also the founder of the 500 Queer Scientists campaign, a visibility campaign for LGBTQ plus people working in STEM careers. She's also the co-director of the Cal Academy's New Islands 2030 initiative. And on a more personal note, I've known Lauren for a long time because we overlapped as doctoral students at City University of New York. And we also became super close friends when she invited me down randomly out of nowhere to Islands and Seas in Baja to help teach some herb course for the nonprofit. And I've been down there multiple times since, living the Baja dream with Lauren, and highly recommend if you want to go herping or just go surfing or just hang out and drink micheladas, which is what I like to do. Anyway, so with that, I'd like to say, let's give a warm welcome to Dr. Lauren Esposito.
Thanks, Sarah, for the invitation and the introduction, but also for the macarons. Absolutely delicious. So today, when Sarah asked my talk title, I admittedly was like, I'll talk about scorpion systematics because that's what I do mostly. And then as I started making the talk, I thought a bit more about what I really want to talk about, and a lot of the projects that my lab has been working on over the last few years have been centered on, and that's really the democratization of natural history and how we try to decolonize the work of natural history and provide opportunity for more and increasingly greater number of people and greater diversity of people to participate in this practice of science. So for most of my career, I've worked on scorpions. I'm a revisionary taxonomist, a systematist, a biogeographer, mostly scorpions in the Caribbean. My PhD was focused on a revision of the genus Centauroides, which is the only genus of medical importance in North America. And up until really recently, that's been sort of my trajectory, my path. Like my path followed a pretty typical natural history museum type of career. But along the way, and especially along the way, visiting places over and over again, like tropical islands, one of the things that I've realized is that I see changes. And I don't know that that was something I necessarily knew I would see as I entered or embarked upon this career. But so I think over time, one of the sort of central guiding philosophy of my lab has been to do faster and to do better, because I've noticed in my short career that things that I used to see aren't there anymore, that they're disappearing. And I think that this quote that was written by one of the other arachnology researchers in my lab, Dr. Sarah Cruz, really sums things up for me in a way that's poignant. And that quote is, because we live during a time when human development is altering the natural environment at an extremely fast pace, we, as natural historians, are often placed in a race to discover, collect, and describe organisms before they go extinct. And this is not a race that we're winning. So my lab strategy is really sort of threefold. The first is to document and describe biodiversity and collect biodiversity data. Like pretty standard, that's what museums do. This is what we're in the business of. The second is really sort of an evolutionary perspective. It's to identify the patterns and processes that have led to the present day biodiversity. And lastly, one thing that's always been really important for me along the way as I'm doing science is to do that science in tandem with education. So almost always I try to set up education opportunities while I'm in the field, especially when I'm in the field working in tropical islands, who may not have access to the same level of biodiversity resources that we do in the global north. but also to train the next generation of natural historians, of biodiversity scientists, but I think more importantly, to train the next generation in a way that is inclusive of people who are from the places where I go to study. And so as I started thinking about setting up this talk, I got to that previous slide, the slide where I say, oh, this is what my lab does. I describe scorpion diversity. And I realized that actually what I've been doing over the last few years is really focused on how we remove disparities in natural history and biodiversity research. And so I want to take some time today to talk to you about some of the things that I'm most excited about that I've been able to be a part of over the last few years. And I kind of broke this up into 3 steps, sort of the 3 steps that we're taking in my lab, in my research group, and at the Cal Academy to make one step forward in removing some of these disparities. And the first step is that we have to demonstrate that the disparities exist. And so I've been doing that in a few ways. First, in June of 2020, during the academic strike for Black Lives, I took some time with some close colleagues of mine in entomology, which, though I'm an arachnologist, entomologists are graciously inclusive of our ilk in their societies. And so we came up with a really simple idea. And the simple idea was intended to solve one small barrier to participation. And that's, how do you know if you want to be an entomologist? So our idea was that we would start a fundraiser and we would raise money to give out free professional society memberships to young people of color who are thinking about entering the field of entomology. Because I think that experience of going to a conference, networking, getting access to the scientific journals that are publishing is really, really important. And it's an important step to deciding whether or not you'd like to pursue a career in entomology beyond your sophomore year of undergraduate studies. So we started this fundraiser. So far, we've raised about $15,000. And we've provided over 500 professional society memberships to over 300 people from all over the world, all people who identify as persons of color interested in entomology. We also started a thing called our Instar of the Month, where we try to feature the people that we're giving these opportunities, these professional society memberships to, as a way of uplifting them as individuals and helping them to network and connect within the entomological community. But we've also been thinking more about what do we know about representation in entomology. And so we started a study based on the National Graduates in STEM survey, the NSF survey that's sent out every year, which is how we track information about who is participating in STEM, who's receiving PhDs in STEM, and what happens to them over time, what that attrition rate looks like. And so what we decided to do was look at the finest grain information that we had available to us, which was entomology and parasitology sort of lumped together. There's no separate category just for entomology. So this was the finest scale that we could get. Again, this is a federal survey. It's been done for years. I think at this point, it's been administered for something like 3 or 4 decades. And what we looked at was proportional representation of men and women across different categories of race and ethnicity that are also collected in this survey. And what you're looking at here is some charts where we scaled the representation to the US national population data. So what the proportional representation is of Black or African Americans, Hispanic or Latino identifying folks, and white folks. On the left side in purple is men, and on the right side in orange is women. And if the circle is smaller than the shaded circle behind it, that means there's proportional underrepresentation. And if it's larger, then it's proportional overrepresentation. And you can see here in entomology that proportional underrepresentation is extreme in both Black and African American populations, as well as Latino and Latino and Hispanic populations. There is no Native American data because there aren't any Native Americans who have been awarded PhDs in parasitology and entomology over the last 20 years of any statistical significance. So what we know now in looking at this data and examining this data for our own field is that we have this proportional representation. It's not something that we're guessing at about when we go to meetings and seeing the faces that are there. It's something that we know based on how people identify in this survey. We can also compare it to sort of broader fields of biological sciences. So for example, here, you can see entomology and parasitology in this dotted box, but beneath it is all biological sciences and all science and engineering. And you can see that proportionally entomology and parasitology are doing worse for underrepresented minorities in science. There's fewer people that are participating, and there's fewer people available to attrition out of that population once they graduate. So when we say that the representation isn't there, it's clear, it's just not there. And so this idea, this entomologist of color that we had, was one way to get over that first barrier to participation for people from historically underrepresented populations. I think sort of the next step after being able to scientifically demonstrate that we have a problem is to acknowledge what our part in that inequity is. And so I've been working on this through a number of outlets. The first is that I led a task force starting in 2020 at the California Academy of Sciences in our Institute for Biodiversity, Science, and Sustainability, which is our research division. And what I appealed to my colleagues to do was to write a statement jointly, acknowledging that not only us as individuals, as part of a collective community in science, but us as an institution played a role in racism throughout history. Sometimes that was an overt role. For example, one of our earliest major collectors in the institution was a eugenicist. And sometimes that was just cultural. It was people participating in cultural norms that were exclusionary to people from historically marginalized groups. So we wrote this statement, we published it. We were actually not given permission by the institution to publish it initially. They were unsure how that would be received publicly. So we published it as a group of individuals on our own on a website, and we came up with a set of 17 steps that we were all committed to taking. Those 17 steps, many of them were things that were individual, like ensuring that we're always collaborating actively with people from the places where we're going to do fieldwork and research. Others were things that we decided that we needed to work on and tackle as a research division at our institution. For example, increasing the number of people from historically marginalized backgrounds in our seminar series. So each of these things were divided up into task forces, separate task forces. And we actually held ourselves accountable. We had regular meetings, and we still publish quarterly updates to how we're doing and how we're progressing for all of those steps that were something we were doing collectively. Some of those have progressed quite far. And I'll give you a couple of examples later of things that we've actually managed to publish as a consequence of this anti-racism work that we're doing as an institution. Another thing that I'm really excited about, that's a paper that's currently in review, led by my master's student, Jacob Gourneau, in collaboration with two postdocs in my lab, Siddharth Kulkarni and Franklin Kalari-Gelme, is a study where we're actually trying to demonstrate what groups are proportionally studied in spiders. I think, like so often as taxonomists, as natural history workers, we're eager to say that things are understudied. It's like a great argument for why you should get funding. But do we ever actually know whether things are understudied or not? I would argue that for spiders, at least, we don't. We have no idea. We have like a feeling that it's understudied, but we have no really quantitative or qualitative data demonstrating that it's understudied. And so what we did was we decided to come up with a metric for demonstrating whether something's proportionally understudied or not. And that metric was really simple. We took the total number of taxonomic entries in the World Spider Catalog, which is the most comprehensive faunal catalog of any group. And we made a ratio that took the number of single changes per species to the number of multiple changes per species, which we interpreted to mean the original description of the species versus subsequent taxonomic changes. And so we said that was a reflection of work on the group. The ratio of subsequent taxonomic changes to primary taxonomic descriptions is how much work has been done on any given group. And we did that for every single spider family. So we took all the species in every single spider family and made a proportional ratio of primary descriptions to subsequent taxonomic changes. And then we categorized every single species, of which there's 50,000 in the World Spider Catalog, by biogeographic realm. My student Jacob went through and categorized all 50,000 species. And we looked at that metric in a number of ways. First, by biogeographic realm. So which biogeographic realm the species, the family generally is distributed within. And secondarily, phylogenetically. So which phylogenetic group the individuals belong to. And the first thing that we found was when it comes to families, we see that there's a really clear divide between global north and global south. And when I say global north and global south, what I'm talking about is the economic global north versus the economic global south. So for example, Australia is a part of the global north, but clearly below this in the southern hemisphere. And we found that of the 5 least studied, according to this metric, spider families, 3 of the 5 are distributed in the global south. And of the 5 most studied, 4 of the 5 are distributed in the global north. So there's a really clear delineation between where the most effort has been done historically working on different spider families in terms of taxonomic effort. And so now we have this metric available for every single spider family. And so any researcher could go through and say, well, this spider family is beyond a shadow of a doubt, understudied or overstudied. And we also sort of looked at these trends through time. And so this is a graph of where we've broken up each group by biogeographic realm. And what you can see here is that for most of the biogeographic realms that are in the southern hemisphere, they've kind of plateaued out through time. Whereas groups that are from primarily the northern hemisphere have really taken this really exponential curve growth to new species descriptions. And so there's certainly been a lot more work being done actively on them over the last 100 years or so. We also were really curious whether there's any particular technologies that have led to major growth in the number of spider species that we've discovered. And it turns out that actually the first illustration published with the spider species description predates Linnaeus, which is when the first spider species was described by Clerk. And the first photograph was in the 1950s and was not of a anatomical characteristic, but rather a web, which is, I think, kind of interesting given that webs have really strong phylogenetic signal in spiders. So the next project that we've been working on, and this is being led by my graduate student, Kate Montana, and it's also currently in review. Actually, it was just accepted 2 days ago, is an archival project. So the Cal Academy has the most extensive scientific archives west of the Mississippi. It was the first natural history museum west of the eastern seaboard. We were founded in 1853. And so as a consequence, we have really extensive archives. And those archives include personal letters and personal effects of basically every scientist who's come through the doors and made that their primary workspace. And so what Kate did is she embarked on this project aided primarily by high school students who are from marginalized backgrounds that live in the city of San Francisco, and looked through the archives looking for evidence of people who have not had recognition institutionally. And that the primary driver of that lack of recognition is personal attributes. They were women, they were people of color, they were maybe never employed, but did the work of a curator or a collection manager. And she took all their letters and correspondence, and her and these high school students wrote biographies for each of these people. It's now being published in the Bulletin of the Society of Systematic Biology, which is a brand new journal launched a year ago. And I think that it's been quite enlightening for me as a scientist, as a modern day curator, to see the extent of the work that some of these people did. For example, Harriet Exeline Frizzell was our first arachnology curator, but she was never paid. She was the first curator of the Cal Academy of Arachnology. She worked there from the 1950s to the 1970s and was entirely unpaid for her entire career, and yet donated all of her life's wealth to the Cal Academy to support arachnology research, which in part supports my current position. So I think sort of the next step along the way is, how do we reimagine what biodiversity looks like today, and biodiversity research looks like today, and who's doing that biodiversity research? And so to do this, we've been working on a couple of things. One of the outcomes of our anti-racism working plan within the research division was that we wanted to reimagine what success looks like in natural history and biodiversity research. The way that people are valued in terms of their quality as researchers. And of course, traditionally, we all know that that's publications and grants. That's the way that we're valued as researchers. We get a number, we get a score, and that score says whether we're good or bad. But the reality is that we all do a lot of things, and those things are in a really broad array. They could be reviewing a court case or providing a survey to a federal government that's a white paper, so it will never see the light of day. It could be mentorship, and in particular, informal mentorship that happens for so many people from marginalized backgrounds that's never counted or categorized because it's not mentoring your primary students. And so we came up with this idea of how we want it to be measured and what success looks like for us, because I think so often success in biodiversity is at odds with what we're trying to do, which is halt the decline of species on Earth. And so if we really value halting the decline of species on Earth, halting the decline of biodiversity, then we should be valuing the things that might aid us in that work. And so I just want to point back to my original quote, which is that we're living during this period where things are declining faster than we can study them. And so if we want to combat that, then we have to be open to recruitment and expand our minds to who participates in the science of biodiversity research. So we came up with these 5 categories. And our idea was really simple. It was that across the 5 categories, you as an individual researcher or a collection manager or a curatorial assistant or a postdoc or a graduate student could decide how you spent your year. And so you would have some sort of pie chart. And your pie chart would say, okay, I spent 75% of my year writing grants and publishing papers. It looks like a sort of traditional 30-30-30 split at an academic institution. I taught a class, I published some papers, I mentored graduate students, and I wrote some grants. Or you could say I spent 75% of my year mentoring and doing outreach. And for each of those categories, we came up with metrics of what success looks like. Some of them are quantitative and others are qualitative. But the idea being that you would be evaluated by whomever evaluates you under the set of criteria that you're saying you're spending your time doing, obviously under the umbrella of whatever your job description is. And we felt like this was a really nuanced but important difference in valuing what we say we value and getting credit for the things that we think we should be valuing in biodiversity science. We've actually implemented this at the Cal Academy. The Cal Academy is a non-tenured institution, so we don't have tenure. We have annual reviews, and so we've implemented this into our annual reviews. And the same 5 categories of criteria are being implemented across all positions. So this isn't something just for curators, it's something that can be applied to really any position within the research division. We also published this in the Bulletin of the Society of Systematic Biology. The next is another paper published in this same special issue of the Bulletin. It was really focused on monographs and how do we modernize the concept of monography. This one was again led by that same student that led the spider study, the spider metric study, Jacob Gourneau, along with something like 40 collaborators from all over the world. And what we really wanted to think about was, what is the future for monography? And how do we need to think about monographs in a way that might be different from how we've thought about them in a past historic sense? In the past, a monograph was something that you did as an individual, maybe in collaboration with others, but really as an individual thing. Like maybe it was your PhD, or maybe it was the thing that you did after you retired and were emeritus. It was something that you spend a long time working on, and it results in this huge body of work. But I think in order to really address, again, this biodiversity crisis that we're experiencing, we have to think about monographs differently. We have to think about them as a living entity, as something that can be updated and changed in real time, not in the course of decades or an entire career even. But also we have to think about who's participating in the process of monographs and who's receiving credit for that participation. For example, I think so often we have field collectors or field assistants who are doing a lot of the work in the field stage, who are maybe forgotten 2 decades down the road when the time for writing the monograph really comes into play. And so really rethinking what credit is and how we apply credit to the practice of monography. And so here you can see in the chart on the right, in the figure on the right, there's this new concept where the monograph is this sort of core central living thing. And at any given time, there's a broad variety of players who can step towards the core or step back from the core, almost like a campfire scenario. And they may step forward during periods of the process of monography, or they may step back during other periods of that process. But all are remaining with focus on the living monograph and the hopes that that monograph can evolve through time and really sort of in real time. So the last thing I thought that I would talk about today is this new initiative that we're just launching at the Cal Academy. And it's really sort of the brainchild of me and Dr. Raina Bell, who's our curator of herpetology. Both of us are island biogeographers, but working in two kind of different parts of the world. Raina really focuses on islands in West Central Africa. I've spent most of my career studying Caribbean islands. And I think both of us have noticed these similar trends. We've noticed that there's a decline of space on islands, they're finite. And as development continues, because humans want to continue their economic development, there's fewer and fewer places for wild things to live on tropical islands. But I also think that tropical islands are bearing the brunt of climate change in a way that we don't always necessarily feel living in larger areas of land, like here in North America, maybe Florida at the moment, actually. And so but I think the other thing that Raina and I really experienced that really stuck with us in our careers was getting the opportunity to participate in education outreach programs on tropical islands at the time we were doing our research. And I think for both of us, even though those experiences were different, they were really transformative and they opened our eyes to a different way of doing biodiversity research. I think, the Cal Academy, like the Field Museum, has a legacy in tropical islands. We have the Madagascar Biodiversity Center in Antananarivo. We've been working in the Galapagos for over a century and have the largest Galapagos collections in the world. We've been working in Central Africa sort of as individual curators. We've been working in the Philippines, but mostly in marine spaces. And I think in each of these cases, there's never been really like the full strength of the institution behind the work that's being done. It's individuals going and doing their research and coming back, maybe with specimens, maybe they left the specimens in some other institution as a deposited material, but it's not the whole academy. And I think one of the most amazing things about being in a public museum, which I hope you can sympathize with being here at the Field, is that there's all these experts that are experts in so many different aspects of communicating, educating, and doing research, all focused on biodiversity. So we're tasked with coming up with a pitch. And this was like something that was open to everyone in the institution. As you might know, or be like, if this sounds familiar, I understand because you also have a new director. But we had a new executive director hired like 3 years ago. And he came in and he wanted to be like a visionary, and he absolutely is. But one of the things that he said was, think big, like think to the moon. Like, how are you going to change the world in the next 5 years? And give me a pitch. And this was a pitch that Raina and I came up with. It was one that I think resonated for a lot of people because it really intended to leverage individual strengths of the institution in a way that was more meaningful and robust, and bring those individual strengths to places that we're already working and that we've already built relationships in. So each of these 5 archipelagos that Islands 2030 is focused on are places that past Academy researchers have worked in for decades. We've been doing, again, research in the Galapagos for over a century. It was really part of our institutional rebirth after the Cal Academy was burnt down in the 1906 earthquake and fire in San Francisco. We have an entire team of experts from the Lesser Antilles, largely arachnologists, who are working in the Caribbean and who have done some of the first faunal surveys of spiders there just over the last decade. We've been working in Sao Tome and Principe in the Gulf of Guinea for over 20 years, and we've led parallel research and K-12 educational programs there every single year for the last 20 years. In Madagascar, we founded the Madagascar Biodiversity Center. It's in Antananarivo, right next to the National Zoo. We've been doing survey work across the island for the last 3 decades. And our history in the Philippines, while it's largely marine, has resulted in some really transformative things, like a new marine protected area that was proposed and accepted by the government of the Philippines just 3 years ago. So the Academy has this really strong track record of working closely with local communities directly. And I think that the outcome is protecting critical habitats in a way that can be transformative for island communities. I think it's something that we're really good at. And I think that when you combine the power of really deep research investments with really deep education investments, what you see is things like UNESCO biosphere reserves being established for the very first time, and that bringing a really huge economic boost to island communities. But up until now, none of the efforts at the Cal Academy that are focused on islands have been led in concert. They've all just been individual one-offs funded by small grants, patching through year to year, hoping that they can get the research and the education done and make it to the next year. So we pitched this initiative, and some of the main motivating factors for why we felt like islands were an important thing to work on is that first, like there's roughly 465,000 islands in the world. I don't think I have to convince you, but islands are really a wonder of geology and evolution. And collectively, they boast the highest concentration of biodiversity that exists on the planet. But they're also epicenters of extinction. And just in the modern written history, 75% of all combined bird, reptile, amphibian, and mammal extinctions have been on tropical islands. And 85% of all remaining reptile, amphibian, and mammal species are highly threatened on tropical islands, along with 50% of all bird species. And I think the biggest problem is that as island ecosystems start to lose their function and balance as a result of this biodiversity loss, the human inhabitants who have been subjugated to intense colonialism and enslavement in many cases over the last 2 to 3 centuries, are also experiencing the loss of livelihood and their ways of life. So this Islands 2030 initiative is really about creating a new pathway for regenerating or restoring ecosystems at scale. Because islands are these relatively simple ecosystems, something that we hold dearly in studying evolution, they're all at risk of these catastrophic cascades of species extinction when just one species is lost. But I think because of their simplicity, we can stop and even reverse the biodiversity crisis on many islands. And there's some really clear data that illustrates what that might look like. For example, this is Choros Island off the coast of Chile. And these are before and after pictures from the island conservation NGO who removed invasive rabbits off of Choros. And it's really just over a scale of 2 years that shows like the remarkable comeback of native vegetation on Choros Island. And so I think that this not only highlights the fragility of tropical island ecosystems, but it also highlights just how resilient tropical islands are when you can start to reverse the past damage that's been done. Cal Academy expeditions to island archipelagos have really been the heart of the institution for over 100 years. Our Madagascar and Galapagos collections are the largest and most valuable in our institution. And they have already, I think, really been used to solidify humanity's understanding of evolution, and within the institution really inspire the public with stories of unique island diversity, what that looks like. We've had major exhibits that were focused on tropical islands. But these collections and their associated data at the Academy are not cataloged. And they're also not available to either the scientific community or the general public. And so through the Islands 2030 initiative, we're aiming to develop a fully open access collections portal where we'll democratize access to all island biodiversity data. And we're going to be digitizing over 1 million records of new terrestrial biodiversity from our collections over the next 5 years. We're going to be using new technologies like the picture array system, which is a conveyor belt-based system to digitize our plant collections. That machine is coming into the building, I think, in the next few months. And we'll be digitizing our entire Galapagos plant holdings in the next year. We're going to be using machine learning to digitize all the old handwritten labels. And that's part of what Picture Array is partnering with us on. And we're going to then use all of that biodiversity data to generate maps of species distributions and richness that we're going to be using to inform where there's still major holes in our knowledge in terms of biodiversity data on each of these tropical archipelagos. We're also going to be using the specimens themselves for DNA analysis. We're going to be doing low coverage genome sequencing of keystone plant and animal species across each archipelago to try to get a real good sense of what the current and past population dynamics are, given the longevity of our collections. For example, the Galapagos collection that predates established human communities living in the Galapagos. I think, really given sort of the inevitability of continued global change, what we fundamentally believe is that we need to stop investing in attempts to preserve ecosystems as they are today And instead, we need to predict and plan for the needs of ecosystems in the future. So through this initiative, our students and Academy researchers are going to be building a new pathway for informed conservation where we forecast that threats using sort of an evolutionary biogeographic framework that we're building. And so in order to accomplish this, we're using genetics to understand the past evolutionary histories of plants and animals. We're combining that information with current land use and predicted climate change models to predict what the future needs of island communities are. And then in partnership with our local collaborators and on-island scientists and governments, we're going to use this information to create island by island models of priorities for conservation. So rather than trying to simply keep things as they are today in a sort of living museum, we really want these island communities to focus on what the future needs of their plant and animal communities are going to be 10 or 15 or 100 years from now. We think that our rich historic scientific collections and these ongoing scientific collaborations will allow us to hit the ground running with this initiative. So we're already underway with this work in the Lesser Antilles. Another huge element of this initiative is K-12 student and teacher engagement, where we're collaborating with local education counterparts in each archipelago to engage island communities in place-based learning and to foster environmental stewardship. For over 10 years, the Academy's been leading a biodiversity education program in the Gulf of Guinea Islands. And this program has had a really clear outcome, and that's the increased appreciation of local biodiversity among residents. One of the most clear aspects of that is that the new currency design for the country, which features native species, includes an Academy photograph of the giant tree frog that's endemic to the island nation. I think that what this demonstrates is that the stories that we've been telling over the last, actually it's 15 years almost at this point, are reaching far beyond the classroom. And in the Islands Initiative, we're building from our prior successes, like in the Gulf of Guinea, to introduce concepts of environmental regeneration or environmental conservation alongside existing educational assets that feature local biodiversity. So many of these island communities are utilizing curriculum that's been developed in either Europe or North America, and or are directly tied to those curricula. And so they're using teaching materials that feature North American or European plants and animals. And so what we're really trying to implement and bring is a better understanding of their own biodiversity so that they have a better connection to it and hopefully as a consequence have more interest in preserving it. These updated curricula are going to be paired with professional development for educators, where we're really bringing the Academy's sort of special brand of environmental learning, which starts with listening to the community, to island communities all around the world. We've already piloted this in the Lesser Antilles with a listening tour of all schools in May. And in 2 weeks, we'll be back in St. Martin, which is an island in the Lesser Antilles, presenting that information to educators from across that northeastern Caribbean region. So that they can start to think about how they would like to modify and adapt curricula that they are already using to include local biodiversity. Alongside this K-12 and teacher engagement project, we're also working with locally based conservation coordinators to amplify the value of biodiversity across these island communities. We're bringing together partners who haven't always communicated historically, from economic and conservation sectors, to local community leaders to devise plans for meeting UN 2030 goals for sustainable development, really focusing on the goals that have been identified as priorities by the UN Small Island Developing States Group. We're also engaging the public at large through targeted social media campaigns that feature and celebrate local biodiversity, and through the creation of public spaces for informal convenings around biodiversity, sort of pop-up exhibits. For instance, in this picture, you can see some biodiversity posters that we produced 10 years ago in the Gulf of Guinea that are still hanging in public spaces and still garner a lot of public interest. And this is something as simple as a poster printed on paper. There's clearly demand and enthusiasm for this information. And I think we're excited about collaborating with a team of local partners. For example, we're going to be installing a biodiversity themed playground in downtown Sao Tome this year that we really envision as sort of a natural venue for organized convenings, for just pop-up exhibits, or just an informal space for community gathering around topics associated with biodiversity. I think for me, the real cornerstone of this initiative and the thing that I'm the most excited about is the creation of a global cohort of graduate students and postdocs who will be trained at the Academy in Biodiversity Science, where they'll gain skills and support needed to become actually a network of conservation and science leaders in each of their respective countries. These teams of early career researchers and their academy mentors will be leading expeditions. They'll be learning new methods, including genomics at our Center for Comparative Genomics, analytical skills in our in-house computing clusters, and the use of tools for digital specimen imaging, ultimately going on to become the world's expert on tropical island plants and animals. The first cohort of graduate students and postdocs is starting right now. They just arrived in the building starting a couple weeks ago and continuing through the rest of the semester. And by the time this cohort leaves and future cohorts leave the academy with either a master's or a PhD, these students will not only be our collaborators, but they'll have a network of colleagues on islands around the world that are tackling similar problems, maybe in different ways. I think the enthusiasm for this has become really evident to me just in the last couple of weeks. Our next cohort of graduate students is from Madagascar. We're recruiting right now for them to start next fall. And after posting about our first info session, we've had under a week ago, we've had over 600 people registered to attend in person in Antananarivo. And so I think that the level of enthusiasm for this kind of training is really quite clear, especially this kind of training that's fully funded. But I think that the impacts of this initiative are really going to extend beyond the islands. And that's because the Islands Initiative is a proof of concept of a new and necessary pathway for how to do conservation. One where we stop trying to preserve current biological communities and instead use scientific data to predict and plan for what's needed in the future where biodiversity and humans live together. Because of the relative simplicity of these island ecosystems, we can really fine tune and tailor our approach with the aim of scaling the lessons that we'll be learning over the next 5 years to larger and more complex ecosystems like those here in the United States. And so I think for me, Islands and the Islands Initiative represent both the legacy of the California Academy of Sciences and our biodiversity research, but also a pathway to a more resilient future for islands, but also for the state of California, for example. So with that, I just want to touch on one final thing, and then I'm happy to take questions. And the last thing that I wanted to mention is something that Sarah mentioned in my opening, which is that I founded an organization called 500 Queer Scientists. It started as a visibility campaign for LGBTQ+ people working in science and science-related careers. And I founded it 4 years ago because I felt like as a biodiversity scientist, I didn't know any other queer people. And it wasn't because they weren't out there and they didn't exist, it's because it's something we don't talk about. It's something that when we walk into the lab and we put on our hypothetical lab coat, that aspect of our identity stays outside of the walls. As somebody who was a graduate student at the American Museum of Natural History, I felt that really poignantly. And then transitioning into a curator position at the California Academy of Sciences in the Golden Gate Park of San Francisco, I was really surprised to learn that I was the first openly queer curator in the institution's 168-year history. And so I started this visibility campaign. It's been going for over 4 years. We've had over 1,800 people contribute their stories from all over the world, from every field of science you can imagine, sort of every stage of career, from retirees all the way through to undergraduates. We have about a 30,000 strong social media community that we try to connect and network. And we get over 10,000 monthly visitors via Google searches to our website. So people are looking for this information, they're looking for this kind of networking, and they're looking for this kind of collaboration. And I think that I wanted to end on this just to say that this is one small way that we can change what biodiversity science, this is extended well beyond biodiversity science, but what it looks like and who gets to participate in that process. So with that, I just want to say thank you, and I'm happy to answer any questions. And I'm sorry for the lack of phylogenies and scorpions.
That's all right, Lauren. Thank you so much. That was awesome. So I'm going to moderate just in the sense I will bring you a microphone because in order for people who are virtual to hear the questions, you need to speak into the microphone. So if anyone has any questions, please raise your hand and I'll come over. Oh, yes, go for it. And then Bruno behind you.
Okay, so you said you had this visionary president who then said like, bring me your ideas. So I mean, this idea, your 2030 islands is amazing. So like, was the institution really supportive? How was it was it hard or easy to get it off the ground?
I mean, I think that it's taken some convincing. Like we were sort of like, it was an uphill battle to take to biodiversity scientists and convince the rest of the institution that this could be really transformative. And I think that one of the biggest points of pushback that we got was like, why islands? And so really like figuring out what the motivation for people who are not biodiversity scientists about why we should care about islands as Californians was like a challenging thing for me personally, I think, as a scientist, because I'm like, what do you mean why islands? Like islands are amazing. Like, have you been to one? But I think that in general, the approach of really including all aspects of expertise from across the institution was one that was appealing institutionally. And so we really endeavored to do that, not because we were trying to like be inclusive, but because we saw those strengths as values, as like really value adds to the kind of science that we're doing and the kinds of things that we would like to bring when we go to communities and do this kind of research.
Amazing talk, so many different initiatives. I'd like to go back to the first one, when the study where you guys are looking at the overrepresentation or underrepresentation. One thing that I noticed is that everyone seems to be at least slightly underrepresented. So what's happening there? Am I who's overrepresented then? If you could elaborate a little more on those results.
Sure.
Yeah, that would be great.
So this like taxonomic effort ratio or metric, I think really what we were attempting to do was sort of assess like what stage we are in terms of science for every family of spiders. And we're like, spiders is probably one of the worst case examples. I mean, I'm sure surely there's like some insect groups that are also terrible and super understudied, right? Like we know that like almost all of the taxonomic record is primary species descriptions and not subsequent taxonomic changes because nobody's ever gone and revised them. We're still like in a discovery stage. But for some spiders, I mean, the fact that the first spider species description predates Linnaeus indicates that there are some spider groups where there's been a really long history of research, and there have been a lot of sort of subsequent revisionary works done within those groups. And so like we were kind of like, I mean, everybody's NSF proposal says that their group is understudied. Like, is that really true? And if it is, like, can we quantify that somehow to direct future research and future investments in spider research? And so I think if we were to do the same thing, to repeat this taxonomic effort metric in a group that's maybe relatively overstudied, like birds or mammals, we would probably see like sort of a different overarching trend where we see like in general that things have approached like completion, so to speak, or have a high level of taxonomic effort that's been done in that group.
I was actually referring to the people who are like even before that. Yes. So I was expecting, for example, in the pie charts to see white males to be like blowing over the pie charts, and there were so what happened there? What because it seems like, if I interpreted right, that in all cases, all your categories were smaller than the pie chart, which indicates
for entomology research. Oh, sorry.
Yes.
I was like totally on the wrong topic.
Yes.
Yes.
So it seems like in ecology, we have an overrepresentation or maybe equal representation to the population of white people. But in all other fields, white is also slightly underrepresented. So what does that mean?
Because what it means is that like this is like proportionally to the US population. So mapping like what percentage of the general US population and what percentage of entomologists. And so like the reality is there's not enough entomologists across all categories, right? So like even white entomologists, like, please come, we need you. There's not enough of us doing the work. But when it comes to like the proportional representation, looking across other demographics, it's pretty clear that that it's severely underrepresented.
Thank you.
Yeah. No, that is like when I first, the person who made the graphic when he when he first sent it over, I was like, I don't know, what does this mean? Like, this doesn't make any sense.
Here I come with the microphone, Kate.
Hi, awesome talk.
Thank you.
Could you tell us a little bit more about some of the digitization efforts, this
Sure.
conveyor belt and this machine
Yeah.
learning? Like
Yeah.
this sounds so futuristic.
Mhm.
Like how are you pulling that? Like, does it really work? How are you pulling this off financially? Like
It really
That's
works.
cool.
And financially, we're getting grant funding. So we were just recently awarded a Gordon and Betty Moore Foundation grant to digitize our California herbarium specimens, and that's what's getting the machine in the building. And because it's in the building, we're able to leverage that and get matching funds through another foundation grant to digitize all the island plants, for example. We've also just purchased a new imaging system for the entomology collections, which is called a Keonce. I don't know if anybody's seen it. It's like amazing. And it takes our rate of digitization of specimen imaging from about 10 minutes per specimen to about 30 seconds. The machine learning for handwritten labels is something that Picture is doing. So that's like proprietary machine learning software, like code that they've written. They're an independent company and they've piloted their machine in a number of institutions already so far. They've piloted it just primarily for herbarium specimens because they're flat, so they blend really easily to conveyor belt imaging. They've also piloted some entomology digitization, but the reality is that the pre and post labor for the entomology isn't really there yet for it to be cost effective for us and our collection and the way that our collection's organized. So for that, we're doing it manually with digitization techs and this new Keonce machine that we were able to purchase with a donor gift. So it's like we're patching it together is the answer. But I do think that, in the past, people have not really been interested in giving to support digitization of collections. And when I say people, I mean foundations, I mean individual donors, I mean all sorts of ways. So how do you fund the digitization of something that people find inanely boring? But I think that the reality is that when you shift the dialogue from we just want to capture the information in our collection so it's never lost to we want to make this accessible to the world, it's really powerful and moving. And that is what we want to do. We want this to be accessible for the world. And I mean, yes, we want to safeguard our collections for the future, but we also want to make sure that everybody in the Galapagos has access to the information that we have sitting in our collection about the Galapagos, because there's no reason they should have to physically access our building in order to get that information.
More questions? Jingmei, do you have another?
So you're putting all this information online, but I've noticed that a lot of institutions are shying away from showing that they have specimens of living organisms just because there's a huge amount of blowback on social media. Have you guys experienced that at all?
I think first I'll say our digital engagement team, which includes social media at the Academy, is really amazing. And they're really good at engaging in dialogue when it gets really hard. Like, for example, people being upset because we have digitized animal specimens. It's not something that we post on digital media constantly, is images of that, but people can access it. We don't shy away from it either. I think it's important to have a team like that supports the work, understands the importance of it, and is really well versed in communicating why it's important so that they can then in turn communicate that to the public. But I think, the reality is we're not doing it for those people. We're doing it for the people that need the information because it's their information. They have a right to that information. And so for the people that are angry about it or upset about it, the reality of the legacy of natural history museums is that people have been collecting for 2 centuries. And we have the information. So should we just throw it in the garbage, or should we digitize it and make it accessible to anybody that needs it?
Any other questions from anybody here? Oh, Bruno.
One more. Slightly related to this, have you come across when you start digitizing en masse and having a closer interaction with the communities where samples were taken from?
Mm-hmm.
Have you come across any issue of repatriation or any kind of conflict that have a reason doing that work?
No, we have a really strong rematriation policy. So what people want their biological resources, which in some cases were taken before there were laws in place to protect autonomy over those, they can have them back. Yeah.
Anybody else? Good. All right, well, with that, we will let Lauren conclude her talk for the day. But if you want to chit chat with her before I take her off to lunch, that would be more than welcome, I'm sure.
Sure.
But otherwise, I want to thank everybody who came in person today because there were good snacks. And also more than anything else, I want to thank Lauren so much for coming and speaking today. Your talk was fabulous. And I
Thank you.
think it really inspired a lot of people here today.
Thanks.
Thanks.